Welcome to the stage, co-founder and CEO of Elemind, Meredith Perry.
Good morning, everyone. It is with great honor and respect that I introduce a true titan in the world of genomics and biotechnology, Dr. J. Craig Venter. He's a visionary in the field of genetics, whose contributions have reshaped our understanding of the world at its most fundamental level. Some have regarded him as the most influential scientist since Darwin, and I don't disagree. Dr. Venter is perhaps best known for his role in sequencing the human genome, a feat that has been regarded as one of the most important scientific achievements in the last century. In 2001, his team at Sorcerer Genomics published the first draft of the human genome, a feat of monumental proportions that ushered in a new era of precision medicine and personalized health. But his contributions do not stop there. Dr. Venter is also recognized for his work in synthetic biology. His team at the J. Craig Venter Institute created the first synthetic bacterial cell, laying the groundwork for what might be possible in the future, including synthetic organisms that could help us combat climate change or even colonize other planets. He also founded HumanLongevity, Inc., a company dedicated to extending the healthy human lifespan using genomics and cell therapy technologies. Dr. Venter has always been driven by the potential of science to improve human life, and his work continues to push the boundaries of what we believe is possible. A recipient of numerous awards, including the 2008 United States National Medal of Science from President Obama, Dr. Venter's impact on science and society has been profound and will continue to reverberate for generations to come. On a personal note, Craig and I have been friends for about a decade. He's been an investor in my businesses, and I'm also on the board of Craig's institute, the J. Craig Venter Institute. And as Craig likes to say, this makes me his boss. So please join me in welcoming my esteemed employee, a man who has truly decoded life itself, Dr. J. Craig Venter. How you doing, Craig?
Great. It's great to be out at sea again. So and it's great to be with Meredith, who's everybody here knows is a wonderful thinker.
Thank you. That's weird as hell. All right. So, Craig, let's begin. You sequenced the first genome in history in 1995. How did that lead to sequencing the human genome and creating synthetic life?
So the first genome was a bacterial genome, and it's something here people would appreciate because it was a new mathematical tool that we developed, a new algorithm for assembling pieces of DNA. So everybody thought genome projects were going to involve thousands of labs and take decades. But what we did is equivalent to taking the Sunday New York Times, putting it through a paper shredder, and then realigning it, reassembling the newspaper. Only we have an algorithm that does that with sequences just by aligning them up. So we shatter the genetic code into little pieces, and then we use this algorithm to reassemble it. So the first genome took a year, it cost about $1 million. But it's an approach that led to almost everything else that we do. The biggest problem, once we had the first genome, so it was the first one in history to be able to look at life from the point of the genetic code, and we couldn't really understand very much. So we decided that the only way to truly understand things was do what Richard Feynman said, if you want to understand it, you have to build it. And so that's when we decided we were going to build our own synthetic genome, trying to design it based on first principles. And it took us 15 years because it was so hard to do. Writing the genetic code, starting with 4 bottles of chemicals, is very complex. And the biggest problem was all the designs we had, not one single one worked. It turns out there are major components for basic life that were not known to science. And so we had to randomly put genes back until we could get a living cell. But it's just like software. We're software-driven organisms, but it's a biological software. It's our genetic code. So once we made this chromosome, around 1 million letters of genetic code, we could put it into a cell that had its genome removed and just boot it up like booting up a new operating system. That's fundamentally how life works. You can't live, and there's no species that can live without the genetic code. So that's a story that took 15 years, but the same approach of using the mathematical algorithms to do the human genome, the bacterial genome was 1.5 million letters. Your genome is roughly 6 billion letters. So the scale of the problem was much greater. Somebody called me and offered me $300 million to set up a company to sequence the human genome. I thought they were joking. I kept hanging up on them, but it turned out they were real. And we put together a team that included the mathematician Gene Myers, who wrote 25,000 lines of new code in 6 months to be able to assemble 25 million pieces of code, genetic code. And we ended up sequencing the human genome in 9 months instead of 15 years using this method.
So talk to us about synthetic life. I think for most people, the idea of creating a new life form is pretty foreign. You've shown us that you can read the genetic code, and now you can also write the genetic code. How do you actually go from writing the code, determining what goes in that code, and then actually building a life form? How does one do that?
Well, it's a complex process, and fundamentally it got down to trial and error, because it turns out 20% of the genes in this minimal genome that we designed, so it's a species that never existed before, we had to keep adding back genes until we got ones that were essential for life. So that's why it took so long. And the rate limiting step is the ability to write the genetic code from these 4 bottles of chemicals. That's changing in the near future with there's a company called Avery that's working out of our institute that is now writing the genetic code on computer chips, where at every pixel they can build a separate chain of DNA. And just by changing the charge at each pixel, they can change the pH and deprotect it so they can add another letter of genetic code. So that's going to change it by 3 or 4 orders of magnitude. And if we had that tool when we started, it would have taken maybe a year instead of 15 years, because there's so much trial and error. We could have built maybe 1,000 chromosomes at a time, and then you screen for life, which one gives you a living cell and which one doesn't. But you need the basic components in a cell or a cell type material that can read the genetic code and make proteins and reboot life. So the cell that we started with to boot up the chromosome changed from the new code that we put in to become a totally different cell, totally different characteristics. And that cell is used around the world by thousands of labs now, because it's the most basic life form and it's the most minimal life form. So it allows everybody to start recapitulating evolution. So by adding genes and processes to that cell, they can add new functions that didn't exist before. So there's a paper just published. This team worked out the genes that it takes for the cell to move around and have mobility. But just think about that. Everything from that cell, if you added enough things to it, we could eventually recapitulate human evolution. But there's a lot of consequences of this. Some have been somewhat flattering. I was on the cover of Time and Newsweek magazine, is this the new God? Both the Pope and President Obama weighed in on the same day. The Pope said Dr. Venter did not create life, he just changed one of life's motors. And President Obama started a bioethics commission to review this entire field. So, I get everything from flattering email to death threats from being accused of playing God. On the joking side, my partner in this work was the Nobel laureate Ham Smith, who discovered restriction enzymes. Those are the molecular scissors that cut DNA at precise points. We always got asked over and over again, were you playing God? And finally, one day to a reporter, he said, we don't play.
We just are. So what are some of the most promising applications of synthetic biology that you're excited about? What kind of life can we make that will change the world?
So it could be the start of a new industrial revolution. Because, for example, when we isolated bacteria from the oceans, most organisms have complex chemistry that our best chemists can't replicate. But by taking the genes and the pathways that make these chemicals and putting them in new synthetic cells, in the future we can make any chemical, any drug, any antibody, any molecules. And there's a whole effort to get microbial cells to be the building blocks for building life outposts on Mars and other planets. So instead of taking the materials there, you just take the microbes and they start manufacturing all the materials you need. So it's basically going to be open to people's imaginations, because with this new ability to write the genetic code, we can randomly screen lots of things. And it's like all of evolution, it's screening and selection versus what we can do just by design right now.
Have you seen other groups outside of JCVI use synthetic biology to create other organisms, or are you guys still the only ones?
So it's turned into a very large field, but no other lab has been able to replicate what we've done because it's so expensive. We had a specialized team of 20 scientists that would be impossible to replicate. And just the ability to do all this stuff, we created all the methods as we went. But there's been a lot of new tools. Most people have heard about CRISPR that you can use for editing DNA and changing DNA. But I think with this new tool that Avery's come up with, I think it'll make it so thousands of labs can start creating new life forms. Now, one thing that I should mention, we think it's really important, we designed watermarks so you can tell synthetic life from naturally occurring life. So we developed a code where we can write the entire English language with numbers and punctuation in the genome. So in the first synthetic cell, we put the names of all the scientists, the names of the institution, and then 3 quotations from literature, including the one from Richard Feynman. So it's possible to write anything now in the genetic code, which is why some people think it would be good for replacing computer memory. But I think DNA is too unstable for that. But it's important to, in the future, that everybody watermarks any synthetic species so we can always tell where they came from.
AI should take note. So talk to us about ethics. The ethical considerations surrounding genetic engineering and synthetic biology are very complex, very multifaceted. How do you approach these ethical questions, and what guidelines do you believe should be in place to ensure responsible research and innovation outside of just watermarks?
Some of these things, it seems simple and obvious. We know the human genetic code. We're learning more and more links to changes in the genetic code, to changes in traits and disease. So it's very tempting to say, well, let's eliminate Tay-Sachs disease from the population by just changing the genetic code in germline so it doesn't exist anymore. And that's a fairly easy thing to do. The question is, you start what's called, one of the things that's called a very slippery slope. Back when USA Today was the nation's newspaper, they took a poll of how many people would use genetic engineering to change the traits of their to-be-born children. And what would they change? And the majority wanted to change fairly trivial things like height, eye color, hair color, muscle types, so they could be distant runners versus sprinters. Very few thought this is a great way to eliminate disease. So getting rid of diseases that kill kids and are very debilitating at early stages is a no-brainer, but how do you stop all these other uses that could have unintended consequences? So CRISPR, everybody thinks it's a precise tool that just changes what you want to change in the genetic code. It actually has what's called off-target effects. It changes all kinds of letters in the genetic code. And so if you think you're using it for gene therapy in humans, it could be changing genes that cause cancer, eliminating key traits, et cetera. So it's hard to start the process on germline without knowing a whole lot more about the genome and having a way to set rules. I can set rules in my lab and what we do. I can't set the rules for what the US does, and we certainly can't set the rules for what's the equivalent of the Wild West in genomics in China and India. So trying to get universal rules where everybody agrees on how we should change the human genetic code, which could improve the race completely. We could find the genes that make Donald Trump who he is and eliminate them from the population. But that may have some unintended consequences that we can't imagine. He'd probably sue.
He might. So the links thing that you just talked about is really interesting. You referenced Tay-Sachs, and we've talked about how just because somebody might have BRCA1 doesn't necessarily mean that they're going to get cancer, and therefore that tells us that the gene itself isn't the whole story. That it might not be a cause of a cancer; it might just be loosely linked to it. So how do we get to a world where we actually do have a full picture of our genomes that we can accurately predict whether or not something's going to happen to someone?
No, it's a great question. And so because my genome was the first one sequenced, hundreds to thousands of labs have studied my genome, and there's constantly papers published telling me how many childhood diseases I should have died from. And genetic studies have been really fraught with oversimplification. So BRCA1 and BRCA2 are great examples that we've discussed quite a bit. If you get them through 23andMe, it's very misleading because they only measure 1 or 2 changes in those genes, and it gives you totally false information whether you have significant changes. But any changes in BRCA1 or BRCA2 at best mean a 50% lifetime risk of getting breast or ovarian cancer, except if you have a family history where every woman in your family has breast or ovarian cancer, your mother, your aunts, your sisters, then the risk goes up into the high 90%, which means we don't know the true causes yet of breast or ovarian cancer, and BRCA1 and BRCA2 are just distantly linked and give hints. So if you get those tests, be very cautious with the interpretation. If you don't have a family history, don't do anything except get frequent checks. From my genome, it was determined that I'm heterozygote for a gene called ApoE4, which supposedly conveys a high risk for Alzheimer's disease. So I'm heterozygote, which means I have 1 copy of that gene. So I asked my neurologist friends at UCSD if I could get a brain MRI and a brain PET scan looking for amyloid. And they said they're not allowed ethically to do it because I didn't have any symptoms. I finally convinced them to do it on the side as sort of a research study, and they only agreed to do it if I would do it under a pseudonym, so that they assumed it would come out positive because of the genetic code. and that I would be tainted with somebody who has Alzheimer's disease, making it harder to raise money or do research. It turns out they called me up and said I have a brain of a 40-year-old. I was 65 at the time. My wife said, no, he doesn't. He has a brain of an 18-year-old. But there was zero amyloid. So it turns out the gene, in my case, predicted nothing. And now we've done thousands of people that are heterozygote and homozygote for ApoE. And very rarely do we see any hints of Alzheimer's disease. So it's probably that gene was a false association that perhaps if you're a boxer, a soccer player, a football player, and you get frequent concussions, then that gene kicks in to help cause Alzheimer's disease, but it's not a general risk. And so there's a lot of misinformation. So if you do 23andMe or similar tests, take everything with a grain of salt. Don't change anything in your life because of that. But now you can get your whole genome sequence for $200, so you might as well get the real thing. But the reports are still limited. We know about 1% of the genetic code and how it really leads to human traits. So that led me to start HumanLongevity, because I realized just the genetic code on its own was not going to get us very far. So at HumanLongevity, we started doing whole body MRI and measuring 2,000 chemicals in the blood, extensive cardiac exam, bone density, cardiac CT. But there's new algorithms developed at UCSD that post-process the MRI data so that tumor cells have a larger nuclei than normal cells, and they have more water molecules in the nuclei, but the water is less mobile. So in the MRI image, that shows up as a bright light. So with this new tool, just reading standard MRI images, they can now tell if you have cancer. So we only wanted to see people that didn't have any symptoms. They thought they were healthy. It turns out 50% of them had something that was significant. And I know a lot of people on this ship have been through HLI, and I hear great stories all the time of what they learned and the outcome. 5% of everybody over 50 has a major tumor that they're unaware of. 1% of everybody here, and everybody we've tested, has a brain aneurysm. And almost everybody knows somebody that's died from a brain aneurysm. That shouldn't happen anymore. It's an hour outpatient treatment to get rid of brain aneurysms versus dying from them. And it's interesting because the data on brain aneurysms was skewed to a much younger population. We rarely found older people with brain aneurysms because they died off. But now it's something that you don't have to die from. And it's a very simple test and simple outcome. So my own life was saved by these tools. I was testing the early MRI machine that we put in, and the radiologist came and told me I had 2 very high-grade prostate tumors. This was after being told that I was completely cancer-free by the best urologist in the world. I had surgery right away, but then I've been cancer-free for 6 years. And that's basically the case for everybody whose tumors we discover using this MRI technique, because we detect them at stage 0 and stage 1, where they're almost always totally treatable and curable. What happens with standard medicine, it's based on symptomology. If your tumor metastasizes to your bones and you have extreme pain, you go to your physician. Physicians are like, detectives. They take symptoms and try and work out what the cause of the symptoms is. But the future of medicine, if I have anything to do with it, will be finding things before symptoms occur. And cancer occurs at every stage of life. It's not just something, they tell people to, when you turn 50, you're supposed to get a colonoscopy. That's one of the things that greets you other than an AARP card. But we've been dealing with 4 women in their 20s with stage 4 colon cancer. So you can't get screened too early. In fact, if you get screened early and they find nothing, it's great, because then you create a baseline for your own measurements for future changes, because we're all a little bit different. And also, if you have that baseline, you know which lifestyle changes are meaningful for you versus this religious approach we take, the lifestyle changes that may or may not help you.
So proactive scanning seems to be the number one approach, at least for HLI, to extending life. It's an expensive thing for people to do, at HLI. But even if you were to just go to go to your doctor and ask for a preventative scan, I don't think that's something that insurance is going to cover. How do we, one, lower the cost, and or 2, get insurance to look at a preventative?
So health insurance companies don't give a shit how long you live. In fact, if you die younger, it's better for their profit. So no health insurance company will invest in any kind of preventive medicine longevity test. Life insurance companies do care. The longer you live, the more money they make, and so they're interested in these approaches. But health insurance companies take the view that every year you can change who your carrier is, so you're not part of the same company long term. So we need to find a way to change that, and change it so that preventative medicine and these kind of tests become readily available. So as you know, we're trying to start a new enterprise for underserved communities and indigenous populations in the US that, if you look at maps of mortality, have by far the highest mortality and the lowest rate of longevity. And we're trying to do it in a not-for-profit world where these people don't have to pay for healthcare. There's now portable MRI units that work in a tractor trailer, and portable clinics. And the Venter Institute is trying to establish several of these to go out and do it, because it should be for free. And the advantage of doing this, the existing databases are comprised mostly of Western European-derived individuals, such as myself, and increasingly Chinese individuals. But if you look at the rate of genetic variations in populations, Africans have the highest rate of genetic variation. There's more diversity within Africa than any other place on the planet. The other extreme are Asian populations that have the lowest rate of genetic variation. It's like they're all derived from the same founder fairly recently. Caucasians are in between the two with sort of a medium rate of genetic variation. So database just of Asian populations or just of Western Europeans is not very useful. In fact, the more diverse the data, the more it would help interpret your genetic code and mine, because it always helps to have outlier populations. And also populations that have been isolated for a long time have very unique genetic characteristics. For example, in Saudi Arabia, they had 12 Bedouin tribes that almost virtually always did first cousin marriages only, and only within their tribes, not even between the tribes. Type 2 diabetes for Bedouins is a selective advantage. So it's a feast or famine trait. And that's why type 2 diabetes is so high in American Indians and other Bedouin populations. But as soon as you switch to a Western diet, it becomes a disease. But in Saudi Arabia and other Mideast countries, they have the highest rate of genetic diseases in the world. They have a huge number of clinics, genetic blindness, diabetes, all the things that occur there. And Abu Dhabi is now trying to encourage their population to marry outside of even the normal population because they've been isolated breeding for so long. But that also creates advantages for finding the genetic link to diseases much more rapidly. That will definitely help these populations. So American Indian populations have a high rate of diabetes, they have a high rate of liver disease, and other things that will allow us to find those bases. But that data and having the broader database sort of helps everybody because without having your physical characteristics and other people's physical characteristics, we won't ever be able to fully interpret the genetic code. So that's where we're trying to take it next, where it should be free, but with HumanLongevity, if it doesn't become a for-profit company, then the experiment fails.
So let's talk a little bit about longevity. We're trying to get people to live healthier longer and live longer in general. What do you see as the upper limits of human lifespan in the near future, and how does that change with our understanding of genomics? So
about the time we started HLI, about 10 years ago, Google started a longevity company. It sort of quickly became an approach that lots of people were taking. And we take a very different approach. My key to longevity is very simple. If we can keep me and you from dying from cancer, from heart disease, and metabolic disorders like diabetes, you'll live a long time.
How long is a long time?
Well, for me, I'm 76. I'm the first member of my father's side of the family to make it out of his 60s. So I consider I already have longevity. HumanLongevity as a company wants to keep me alive for a very long time because it's bad for business if the founder dies early. I'm trying for 100 or more, but I've changed my characteristics. One of the things the MRI measures is organ fat, particularly liver fat. It's one of the best predictors of disease and lack of longevity. The only way you can get it measured is basically with an MRI test. And I was on a 30-mile bicycle ride with a world expert on organ fat. And we stopped for a break at 15 miles. And he said, the only way you can reduce organ fat is with calorie reduction. And I said, why in the hell didn't you tell me that 15 miles ago? Because I was thinking exercise has to do it. But only by calorie restriction can you change your organ fat. So I've lost 55 pounds in the last 12 to 13 months, and my organ fat went from very high to very low levels. So just by changing your weight and changing your metabolism, you can change your longevity, because people with high organ fat die sooner than later from liver disease or need a liver transplant. So you can take control and you can totally change your outcome, but only if you have data. We don't know what the upper limits are. Clearly there's these golden populations that it's not unusual to live beyond 100. So genetics, good genetics clearly play a role, but is it just good genetics that does what we're trying to do? It prevents heart disease, prevents diabetes, prevents obesity, et cetera. So it's not clear that there's an upper limit. At some stage, things have to wear out, but we're learning how to replace things. But you talked about ethics earlier and asked me about ethics. Under the current conditions, I think it's unethical to try and get people to live to be 150 or 200 because we've already overpopulated the planet. We're destroying the planet because we can't sustain the population that we have. So if everybody lives to 150, we will greatly increase that problem. and the population has to go up unless we stop reproducing. So while there may be no upper limit, we should have, like there is with your iPhone and automobiles, planned obsolescence to make sure we don't get the whole population shifting to 200 years old.
Wow. So you think that if we were able to live forever, that there should be basically like a law on how long someone should be able to live?
Well, or have mandatory sterilization after a certain age. So,
Sure.
We don't have 150-year-old men fathering kids.
I like that a little bit better than the planned obsolescence. I'm not sure I feel so great about that.
Well, our genetic code has planned obsolescence built in right now. So we have to alter that either by altering the code or severely altering lifestyle.
So what I don't understand is that, trees can live for thousands of years. We've got tortoises living for hundreds of years. As I mentioned yesterday, there's a clam named Ming that has lived till 506, and Greenland sharks that live until like 500. What is different about these species than humans, and how can we take some of these specific genetic traits that contribute to longevity and integrate them into humans?
Well, tortoises, like the tortoises in Galapagos, move very slowly. They have sex for a very long time. Trees move even slower. So there are things with different genetic traits and populations that show that early death is not mandatory. But look how longevity has changed just in the last 2 centuries. People living into their 50s and 60s used to be very much 200 years ago the older population. Before that, it was 30s and 40s. So by changing infectious disease rates, having vaccines, having antibiotics, having healthier environments, and then now switching, or people trying to switch to healthier diets and preventing disease, we're starting to change the equation. Eventually, we will change your genetic code so you'll have better traits and live longer. To me, what's important is not the length of life, but the quality of life. We can have people live very long time on respirators and recirculating their blood, even if they're brain dead. That's kind of a poor quality of life, I would assume.
So other things that you might not know that Craig has done, he's done so many incredible things in his career, but he's also done work to genetically modify pig organs to transplant them into humans. And the first person with a genetically modified heart was implanted, I think it was last year, was it?
Yeah. And that person lived longer with we call that humanized pig heart, lived longer than the first human heart-to-heart transplant. So this is work by our friend Martine Rothblatt. She had the idea because her daughter has a lung disease and she wanted to find a way to continue to save her daughter's life. And she thought by changing the genetic code in pig lungs that it would provide a future source of lungs for her daughter. She came to me and asked if I would start a team to re-engineer the pig heart, and we put together a team, and it was absolutely amazing to me. We only changed 10 genes, and it immediately changed how long these human-modified pig organs would last without rejection in baboons and now in humans. And Martine's company is about to start a major clinical trial doing a very large number of heart transplants. People heard yesterday how many people need heart transplants a year. And so having a perpetual source of organs would be fantastic. Kidneys are also in the work. About 1 million people in the US need a kidney transplant a year to live. Kidneys are a little bit more complicated because we have to alter some of the pig gene metabolism, but I think that'll be readily easy to do. I suggested also using the humanized pig skin for burn patients, but everybody's worried they'll start liking football more. But there's unlimited use. We already use pig heart valves, and because they're fairly inert, they're not rejected. But as we change more and more genes to humanize the pig heart, hopefully we'll get to a point where they can work without using anti-rejection drugs, which are pretty severe for people to take constantly. But Martine is the one who really deserves 100% credit for financing this, thinking of it, getting, it's very complex. We made a new pig cell. Her teams turned that new pig cell into a whole line of genetically modified pigs that to produce enough organs, obviously you have to grow large numbers of these. So the work that's gone into it is pretty amazing and will lead to longevity for at least the people that are dying from need of heart and kidney transplants in the near future.
Do you believe it's possible to indefinitely replace organs to extend life?
For most organs, yes. Replacing your brain sort of doesn't do you a whole lot of good. But I think one of the biggest findings of the last 20 or 30 years is that most breakthroughs in science are overcoming the ignorance of the past, not just because they're great discoveries. So scientists and neurologists have assumed forever that your brain cells don't grow or change throughout your life. And then it was discovered that brain cells are constantly growing and changing. It's called neuroplasticity. What you do in your life, what you're exposed to, can change neuronal circuits. And so the brain can always adapt and change if we give it genetic opportunities so that you don't get amyloid plaques and other things that cause neurodegeneration.
So let's switch gears for a second. You've been involved in various efforts to explore and catalog genetic diversity on Earth, such as the Sorcerer Expedition. First, tell everybody what the Sorcerer Expedition is and what is different about that expedition from Darwin's expedition with the beagle.
So the same mathematical tools that we use for assembling the genome, it made a discovery by accident early on. We were sequencing a clinical isolate from a patient in Norway, and the algorithm doesn't make any assumptions. It just assembles what's there uniquely. And it turns out this patient had two totally different strep organisms, but the algorithm totally, instead of mixing them together, assembled them so we had two separate genomes. So that was the beginning of my thinking that every human on this planet and every species on this planet has what I call a unique mathematical solution for their genome. We could grind you up, sequence all your DNA at once, and the computer would assemble your genomes back into your unique genome. So I had to decide to apply this to something I've always been interested in, and it's ocean life, which the oceans around us gives us about 60% of the oxygen we breathe. It's responsible, these organisms are responsible for all evolution of higher life. But scientists, again, thought there was very little diversity. So the seagrass you see out floating in the ocean is Sargasso seagrass. So this is called the Sargasso Sea. And it was thought to have the lowest microbial diversity. So we just took a sample of 200 liters of seawater, filtered out the microbes, and sequenced all the DNA all at once. And it turns out we discovered, just from the first experiment, over 1 million new genes and about 10,000 new species. So this altered the thinking of the environment drastically. And we thought if we took samples in different places, there might be populations that were more abundant in different areas. And so we could sort of start to assemble the ocean microbiome. One of my dreams was always to sail my own boat around the world. And people think my best idea was to sail my boat and do science while doing it and get paid for it. So we started the Sorcerer Expedition. Sorcerer was a 100-foot sloop. We started sailing around the world. We did 65,000 miles and stopped every 200 miles to take samples, filter the seawater, and ship it back to the lab to sequence. So that expedition discovered about 100 times more species than the entire history of science has so far. In fact, we've recruited physicists and astronomers because we've shown that there's several orders of magnitude more unique organisms in the ocean than there are stars in the universe. So when you look up and see all that diversity, look out here in the water, you can't see them, and there's far more diversity there. Darwin couldn't see it. Darwin did things based on his visual acuity. He could see plants, he could see animals. You can see the most crystal clear waters in places in the French atolls and stuff. 100-foot visibility, it's crystal clear water. Everywhere on this planet, every cc, every milliliter of water has 1 million bacteria and 10 million viruses. So if you go to the beach and you take a mouthful of seawater today, think about that. But these viruses and bacteria actually keep us alive by making oxygen. They're the basis of the food chain. We have a book coming out in September with Harvard Press on the Sorcerer Expedition. It wasn't just fun sailing and fun science. Geopolitics entered into this because every country owns 10 or more miles offshore of all the species and diversity. So we had to work through the State Department and every government just to take 200 liters of seawater if we were in their waters. And some countries wouldn't let us do it at all. We got arrested twice. We got arrested by the French in French Polynesia because the French government had a debate with the Tahitian government of who controlled who could do science there. The French government wanted only French scientists to make discoveries. They didn't want this American scientist to do it. They threatened to sink us if we left harbor. The State Department and the U.S. Navy had to intervene and say that we were a U.S. research vessel and under protection of the U.S. Navy. So we're also arrested in the Indian Ocean by the British because they thought our notoriety would bring attention to Chagos Island and the islands there where the U.S. and the British gathered up all the natives and kicked them off their islands. And they sue every year in U.S. court, but we rent Diego Garcia from the British. That's our biggest B-52 base in that part of the world. So even though we notified the State Department that we were going there, they sent a military vessel looking for us, arrested us, took our passports. But we finally negotiated our release because I had a satellite phone and my friend was the British ambassador at the time. And so I just had to sign that we broke a law from the 1800s of having unlicensed dive equipment aboard our sailing vessel. And we're eventually allowed to leave. So you find a lot of exciting tales in there, but it's some of the most important science because every day we are changing these microbes. just killing off one from a 1 degree temperature change in the Indian Ocean has made all those beautiful white sandy beaches you see in the Seychelles. But they're from this one bacteria dying, and therefore the coral was dependent on this microbe. The coral died, and now you have all these white sandy beaches. So as we change the temperature of the ocean, as we fill it with microplastics, we're changing the environment that keeps us alive. And sooner or later, we'll destroy that if we don't change something. So there's a lot of science, a lot of sea adventures, and a lot of geopolitics.
So definitely make sure to get his new book, The Voyage of the Sorcerer 2, in September. I had the privilege of reading a preprint, and one of the major takeaways for me is that life is almost infinitely diverse. There's a huge amount of diversity between the sequences of any given gene. Why do you think that there's so much diversity?
So we initially characterized life as a species based on the conditions we live in. But we have microbes that live in pH 11. If you stuck your finger in that water, it would dissolve your skin. There's ones that live in strong acid conditions and the acid mine runoffs. There's organisms in the deep oceans that live off of metabolizing sulfur and CO2 into methane. There's more life forms and diversity on this planet that most people don't know it, but we exchange about 100 kilograms of material each year between Earth and Mars. And that's before we started sending up spaceships and things to contaminate it further. So you can't take a shovel of Earth on this planet without having some Martian soil in it, and vice versa. So I am certain that the basic elements of life, and we find hints from asteroids, we find the nucleic acids that form the genetic code, we find amino acids that are the basis of proteins. These are going to be ubiquitous chemicals everywhere in the universe. And everywhere we have conditions of life, now for higher life, as far as we know, we need oxygen. But for other types of life, they can live in very extreme conditions where we wouldn't survive even for a few seconds. So microbial life, I am certain, will be ubiquitous anywhere that we can get to and look. We're a very young planet, so whether there's going to be evolution to higher life forms elsewhere from the early microbial life remains to be seen. But, you know, with the water under the surface of Mars, it used to have oceans like this. I am certain that we will find abundant life on Mars and history of life on Mars that won't look any different from the diversity of organisms that we find here. So I'm certain life is ubiquitous and will all be based on the same software code of DNA and RNA.
Why do you think it's going to be based on the same software code?
Because we find the same molecules ubiquitously, from asteroids that have come from great distances. We find the basic chemicals of life on those asteroids. So the basic building blocks are the same as far as we can tell everywhere.
So you see the basic chemicals of life, but you haven't found life on asteroids.
Yeah. So, people have, hypothesized that you can get silicon-based life. You can only get that where you live in Hollywood with plastic surgeons.
So we talk about water being a critical component of life on Earth. Do we think that water is necessary for life everywhere, or does it just happen to be necessary for life on Earth, and that might not be true for all life everywhere?
So the microorganisms in your gut, in your intestine, there's tens of thousands of them. Most of them are what are called anaerobes, means they live without oxygen. There's not a lot of oxygen in your intestine. And so anaerobic life clearly existed before there were oceans and before there was the photosynthetic organisms to create oxygen. So life does not need oxygen to exist. Mammalian life and the visible world that we know is totally dependent on oxygen. The invisible world is can live on all kinds of substrates and doesn't need oxygen. So if we have oceans, I'm sure we'll find life and photosynthetic life, but I'm sure we'll find life. We took samples from a mile deep in the earth. They're all anaerobes. They live off of different chemicals, and they live at very high temperatures. So the highest temperature is something like 160 degrees centigrade. We would dissolve very rapidly, drop into those temperatures. So, NASA is very good at now looking at extremophiles instead of just looking for life that mirrors human life.
So we probably only have time for one more question. So I'd love to understand from you what's next for JCVI. What's the most exciting research that you're working on, and what do you hope to accomplish in the next decade?
So everything I've done in my career, from sequencing the first microbial organism to the human genome and discovering this diversity, has been trying to understand the genetic code. My dream is to really be able to truly understand the human genetic code and predict things. And we're so inept in this space. So simple things like wiring of our brain. So inbred mice, the neurons show up in exactly the same position, plus or minus a micron or two. So the wiring in our brains is clearly genetically controlled. And so some people, probably a lot of you, have good photographic memories. Every summer for a long time, I spoke to a select group of several thousand high school students, roughly 20,000 of them, in a soccer stadium outside of Boston. And they're selected as the best students in the high schools around the country. So I have what's called aphantasia. If I close my eyes, I have no visual memory. I can't see pictures. But our education system is based on selecting for people that have photographic memories. So I asked these 20,000 students how many of them thought they had photographic memories. It was essentially close to 100% of the audience, because that's what we select for. We select for rote memorization and regurgitation. I asked how many thought they had aphantasia, and there was only maybe 100 or so, because we're selected against in the education system. But somebody like myself with aphantasia is great to have as a leader of an organization if they can survive long enough to get there, because I think only in concepts, and I can meld all kinds of concepts together, and usually in an area like sequencing the human genome, I'm the only one that believes the idea for a long time. So there's advantages for each, but they should be detectable simply from a genetic test or an MRI test, and think about changing the education system with individuals not only with different learning disorders, which would be predictable, but with aphantasia should be in a different track than schools based on rote memorization, just as one simple example. But think of all the complexity of the brain. We should be able to understand that wiring by looking at the genetic code of populations of people with vastly different traits. And there's not, there's probably 1,000 different types of intelligence and brain structures that we're so primitive right now, we're thinking we have no idea what they are. So the hope is by doing genomes and the MRI and other phenotype data on diverse populations, including unique populations with human traits, we'll get to a faster understanding of our own genetic code. So That's what I hope to spend the next 25 years working on.
So as a final note, JCVI is responsible for some of the greatest achievements in science and healthcare in the last century, and your research has probably saved many lives of people on this boat alone. Without your work, genuinely, we would all be worse off. And as a board member, I intimately understand that this work is only sustained by donations and research grants. So if anyone would like to get involved, we'd love to hear from you. And the world and you yourself will probably definitely benefit. So Craig, thank you so much. You're an absolute genius.
And thank you, Meredith. You're a great boss to have. So.